Analyzing your samples in the TN-TOC is only the first step in getting your results. The raw dataset will still have to be formatted and organized using pre-made Excel tables. Sophia, our lab manager, will demonstrate how to convert raw data here, using a student's results as an example.
What I have up here is our TOC-TN data analysis template sheet. The first tab here that I have open is our raw data tab. So I'm going to take the data that we got from the TOC, put in our raw data tab, and then I'm not going to touch it or manipulate that data at all. You always want your raw data pristine and untouched. But what I'll do is I'll grab that data and put it into another tab that I'll start manipulating it. So let's open your raw data from the computer, which is in the form of a text file. So in Excel, you want to open your text file, but what you need to do is when you click open, it says all Excel files. I actually want all files because it's a text file. And so as you can see, it's not here because it's only opening Excel files, but you have to go here, say all files, and then it turns up.
Nice.
So you want to click open, and then it's going to ask you some questions about how you want to import it, but you just keep clicking next until you can't anymore, and then you just click finished, and then here is your raw data. So I'm going to copy this, and then paste it into my raw data tab. And I always delete this data because this word data because it kind of screws something up later. But anyway, so this is your raw data. So the first thing I'm going to do is I'm just going to grab my table here and
how
copy
do you do that?
it.
Sorry to interrupt. What key do you press?
So I click sort of anywhere on the data and I push Control A once, and it highlights my table. You want to be careful with the Control A function because depending on how many times you click it or where you're what cell you're highlighted, it sometimes highlights the entire spreadsheet, sometimes just the entire table. So, you know, if I click Control A twice, it actually highlights the entire sheet, and that's what we don't want. I just want this table here. So Control A, Control C, and then Control V.
Sorry.
Okay, so then all of the sample names should have a unique identifier. Your specific, Connor, data here, I believe, since we were doing two sets, there is a duplicate sample name starting here that we discussed. So what I'm actually going to do is those do need to be renamed, and so if I just do an _2, would that make sense to you?
Is there a way you could merge the cells together?
Would you prefer that? Yeah. And so what you do to merge cells, you go equals, and then you click on, I'm going to click on this cell, and then and then a quotation mark, and I'm going to put a space and maybe a parenthesis. And then a quotation mark, and then the date, and a quotation mark, oops, this thing, quotation mark, enter.
Wow.
Oh, actually,
the
dang
format's wrong.
Yeah, it's not gonna, so, okay, I'm just gonna edit this, so then I'll just manually type it in. There.
That's really cool.
Perfect.
Yeah.
So then I'm just gonna drag this down. So there. That should be good. Copy that. And then what you'll want to do is paste as text, which is this one, or just the values. Otherwise it'll paste.
And be like, what's going on?
I'm just going to delete this. I forgot to mention, so what this raw data is, or this is the raw data, but what the data is actually it's going to give you one line per injection per analysis. So all of these I'm highlighting here, this is all from rinse 1. So what you can see here is it did 5 injections for carbon, but this is the mean area, mean concentration. And then it gives you the nitrogen data, mean area and mean concentration. And so what I'm going to do here is I'm going to tell Excel to delete all the duplicates of sample names, which will leave me 1 line of data per analysis. So the data is in the raw data TOC tab. I'm going to do Control A, and what's important is you do Control A once, because it just highlights my table. If I do Control A twice, it highlights the entire sheet. And then what I'm about to do, if you highlight the entire sheet, it will actually like shut down your Excel file, which you don't want. So Control A once just to highlight all of the data. Then under the Home tab, under Styles, you go as Format as Table. And I'll just pick the first one. Make sure My Table Has Headers is checked, and you click OK. So then it's formatted your table so you can sort of sort through the data. The next thing you're going to want to do, I'm going to zoom out a little bit. Under the analysis, I want to separate the carbon and the nitrogen. So in analysis, I'm going to just sort A to Z. So it's going to put all my carbon first and all my nitrogen second. So what I'm going to do is I'm going to scroll down and find where my nitrogen starts, which is here. And then I'm going to highlight all the nitrogen data. And then I'm going to cut, this is important, make sure you cut, not just copy, but cut. And I'm going to paste the nitrogen data into the raw data TN tab underneath my titles. Perfect. I'm going to click Control A, which is going to highlight my table. I'm going to format as table, the nitrogen data. And there we go. And then now what we can do is tell Excel to delete our duplicates. So I'm clicked on my table. If you go to the Design tab, under Tools, you click Remove Duplicates. And what you want to do is unselect all and only select sample name. So I'm saying Excel, only delete the duplicates under the sample name column. And then you click OK, and it deletes all your duplicates. It says 852 duplicates were found, leaving 124 unique values. And so I click OK. Great. So, perfect. Delete this last line too. And so then I'm just going to do the same thing for the nitrogen data. I'm going to go to the Design tab under Tools, Remove Duplicates, unselect all, only select the sample name, and click OK. So there we are. And so what we've just done is probably hours of work manually deleting all the duplicate lines to get our data. So from here, I'm on the Raw Data TOC tab. What you can do is you're going to copy that and paste this text onto your TOC data analysis sheet. I'm going to go back and I'm going to grab the mean area and concentration here, copy, right click, paste this text, and then I'll just grab the file numbers just so we know.
So why would you take the mean area if you already have concentration?
Because the concentration is based off of the area. And so if at any point in time you actually wanted to recalculate, you wanted to delete a point in your curve and recalculate the data, you would need the mean area to do that.
Okay.
Then you can recalculate the concentration.
Interesting.
So, and the other thing I should note is what I do is I keep track of the curve, the mean area data for the carbon curve that we ran last time. So every time you open this sheet, you need to check on the computer what the latest or what carbon and nitrogen curve your data is based on, because I run maybe once every month or every other month a new curve. So this is going to be constantly be updated, and so I always write the date that this curve is. So you'll just need to check that on your own every time we run it. And then so you do the same thing for nitrogen. grab all the sample IDs, TN data, your mean area and concentration, and grab the vial numbers. Great. And so, and you can, now that we have all the data in here, then we can start copying and pasting our rep IDs, our spike IDs, and then start inputting all the data into our QC charts here. If you need to add some more rows, you can do that to this. So, for example, our carbon QC, I just push equals, and then I grab the final concentration for our first carbon QC, which is here. And what it's going to do is it's automatically going to calculate your percent accuracy. And it says here where the range that your percent accuracy needs to be in. And then from, so then you can determine whether or not that QC passes. And so you just go down the line, you push equals, you grab your data from here, and everything's calculated for you. And if there's any issues, you can see that and mitigate that there. The spike is calculated for you. The equation is here.
Unless, of course, it's
And
a different spike.
so you'll need to edit this to whatever, you know, we don't do the exact same spike for every single sample we run the instrument. It's totally based off of what your samples are and what the spike needs to be for your samples. So you'll need to edit this to fit your samples, whatever it may be.
A couple quick tips. Firstly, analyze your raw data as soon as possible, as you may forget important dilutions or changes to your design. If you analyzed only total carbon and total nitrogen, you can ignore process replicates required for MBC in the standard section of the Excel table. Finally, keep a lab booklet to keep track of your raw data and processes, as this will help clear any confusion down the road with your results. And with that, you should have the tools to format and extract the data you need for your experiment.